Good morning, everyone. So today our group will be talking about the gut microbiome and its relations to depression. My name is Spencer Gout. These are my colleagues Alex Meyer and Emma Scaff.
Sorry.
All right, so let's get started. So over the course of the week, we've been addressing a few different types of imaging techniques. But more importantly, we have looked into how they can be applied to real life. So our group today, we took interest to how microscopy can be used in the context of medicine. And today, the medical challenge at hand is a serious and worldwide disease that affects many. So we will be addressing the broader challenge of the microbiome using microscopy. So currently in the gut, there's a family of bacteria called Lactospiraceae. Lactospiraceae are known to have a positive correlation with major depressive disorder, otherwise known as depression in humans. So this correlation is known, however the causation isn't known. The functions of the bacteria, so we don't know this, and that's what's being left out in the open for us to figure out. So whether this is on the chemical basis of a neurotransmitter being inhibited on like a neural synapse, we really don't know yet. So that's what we're trying to figure out. So in our experiment, we plan to use fluorescence microscopy to monitor the bacteria and the relationship with neurons in the gut, so to further understand our gut's microbiome and how it affects our central nervous system.
Wait. Do you just want to hold this?
Yeah.
Sorry.
So we will use this information to determine if neurons and their relationships with bacteria elicit depression itself. So we chose fluorescence microscopy. So for this project, we chose fluorescence microscopy especially. And I think Dr. Bentolila said it best when he said that the main goal of fluorescence microscopy is to derive an image based on the interaction of light and matter. So this is done by passing forms of light, say a laser, and photons being passed through a series of lenses and mirrors until a proper image is produced. And with this image, researchers can analyze and further studies and conclusions. Now, there are two major advantages to using fluorescence microscopy in the context of our experiment. One is that this technique is non-invasive and will not damage our cell cultures. And secondly, its resolution is strong enough that it will be easily able to see the neuron functions. So as I said earlier, fluorescence microscopy can be used in the gut microbiome. So pictured to the right is a spatial organization of various gut bacteria. And pictured to the left is a three-dimension landscape of the gut's microbiome in general. And these were taken with fluorescent microscopy techniques. So I'll pass this on to Alex, who will talk about the details of our experiment.
Okay. So with what we currently know now, there are several groups of bacteria that reside within the gut microbiome that that relate to depression. Sorry. But what we don't know, like Spencer said, is that we don't really know like the importance of which ones have like a greater effect at causing depression, and we don't quite know the specific functions and how each of the bacteria like cause depression. But despite knowing like there are a whole bunch of these different types of bacteria, we're specifically focusing on the bacteria in the gut microbiome that fall under the Lactospiraceae family. Due to research that has been previously done, we are aware that there is a positive correlation between having these types of bacteria in your gut microbiome and the chances that a person would experience the symptoms of depression. We currently do not know like what mechanisms allow for this to happen though, and that is what our study is trying to figure out. So specifically, our study is trying to figure out if the mechanisms that cause depression are due to direct relationships between bacteria interacting with the neurons in human guts. So to run this study, we are going to first grow neuron cells in cell cultures. Then we are going to introduce bacteria that I've previously mentioned. But in order to be able to see anything going on, we need to encode these bacteria with genetically encoded calcium indicators, or GECIs. Just like how fish can be encoded into bacteria and works as a fluorescent marker for proteins, GECI can also be encoded into bacteria and works as a fluorescent marker of calcium 2 ions, which would help us see if there is a relationship between the bacteria and the neurons. And the use of light microscopes allow us to see this happen in real time. And I will let my colleague Emma explain the importance of what our findings may be.
Depression is a significant problem in the United States, seeing as it affects 7.1% of the entire population each year. Through determining the gut microbiome's relationship with depression, solutions can be discovered in order to hopefully reduce the amount of affected Americans, as well as people worldwide. From the setup of our experiment, the relationship between the gut microbiome and depression, we can deduce that there will be one of two outcomes. The first case scenario would be that the results show a definitive connection between the microbiome and the disease. Through the results involving calcium ion channels, we will be able to see the bacteria being used interacts with the neurons in the cell culture. Thus, the connection from the gut to depression will be proven. Therefore, a reliable clinical treatment can be found based on how the microbiome proves to affect the body. The second of the two outcomes is that there will be no feasible correlation between the bacteria in the microbiome and depression. These ambiguous results will be shown by the bacteria simply not having any interactions with the neurons, proving the lack of connections between the two factors. If the experiment does not display the predicted effect, the outcome will still be able to contribute to the research field of the causes of depression. By ruling out the possibility of bacteria from microbiomes interacting with neurons, focus can be pointed elsewhere, putting researchers closer to the end goal of finding the gut microbiome's overall effect on the body. No matter what the final outcome of the experiment is, it will undoubtedly help to progress the research community, whether it be by finding a new connection to assist in finding treatment or permanently ruling out something that was once thought to be a cause of depression. The data found will be able to, in some way, further research surrounding the gut microbiome. Do you have anything after this?
So during the Q&A, if you guys could please repeat the questions because they are not mic'd. So for the people who are watching, it's helpful if you can see them. So
Any questions? Yes.
First of all, congratulations, a very nice presentation. It's the first one, so I know it's not easy to be the first. My question is to you guys, like, from what you have been reading, you mentioned like a positive correlation between that specific bacterial type and depression. And my question is, do you know if that specific bacteria is present normally in the human microbiome, or it's just not present at all, and this is when you have it, but you show signs of depression?
That's a great question.
Repeat the question.
Oh, yes. So is the bacteria that we're looking into, is it found in like humans without depression, or is it like just found in reduced amounts? From our research, I'm pretty sure it's just that there's like an abnormal amount of it, like there's an increased amount of that type of bacteria in human gut microbiomes. But yes, sorry.
As a follow-up to that, how would that help you decide what to do about that? For example, would you want to, for example, kill all of those bacteria?
So the question was, how would that, like, how can we take understanding if this is present in all people, how can we, like, create a treatment for that? Any of you have an idea of this?
So I think if there's like if a connection is found, say because serotonin is a neurotransmitter correlated with happiness in the body, so a lot of what depression does is inhibit serotonin from binding onto the neural synapse. So if a relationship was found with the neurons and the bacteria, then we could possibly deduce with further research that the bacteria are likely producing some sort of substance that attaches onto that receptor, onto the neural synapse. So what could be done with clinical treatment is to target, using antibodies or some form of that, is to target what is being inhibited on that receptor site. So that's one thing that we could do about it.
Can I say one more thing on that? I don't think completely killing off that bacteria may be entirely beneficial, though, because like a regular biome with animals, there are specific niches that each of these bacteria fill within you. And I think if you kill off all of one type of bacteria, another harmful bacteria is allowed to come in and replace it. It's like what's happening when you take antibiotics. You're killing off all the bacteria, thus allowing for much deadlier things to come in and affect you worse. Yes.
Another question. I really like the way you break down your experiment, right? Trying to really outline how you would do the experiment. So you explain how you would mix the bacteria with a culture of neurons and how you will use the optical microscopes to look at them. So my question is, do you have any idea of what you would be looking for?
To just start off, I think we would want to be looking for like just an indication that like the bacteria is sending out calcium ions in some sort to like activate the neurons. But if we find that relationship, then more can be done to look into like what specifically is going on with the bacteria, because if they're not doing that, then there's no point to like look for more. So this is just the starting point, and more research can be done after that.
And so to add on to what he said, I think more specifically what we'd be looking for is like a presence of the lacnospirosate in between the synapses, right, in between the neurons. Because like I said, serotonin, and I'm sure there's other neurotransmitters that are correlated to depression, but the basis of this would be looking for specific inhibitory responses to serotonin. So we'd be looking for a presence of that between those neurons.
So functional activity represented by calcium signaling, and then spatial location to see if they're in one particular sensitive location of the nerve.
Yeah, and that could be, motor neurons, inner neurons, or sensory neurons, but further research should be done.
I see a student question.
Oh.
Oh, good.
Yeah.
How are you planning to use calcium 2 plastosis in your research again? Like you mentioned that you would coat bacteria with calcium 2 plus.
What? Oh no, we're not going to code it. Oh, sorry. How are we going to use calcium 2 ions to help in our research? You said that we are going to code it. No, we are going to encode. Sorry, we are going to encode things called genetically encoded calcium indicators, which basically attach fluorescent proteins to calcium ions produced by bacteria, so that you can simply like track them. And neurons work by having calcium ion gates that like start transduction. So we're just looking for that at first, if that answers your question. Okay.
All right. Thank you, guys.
Yes.